Welcome to part 3 of 3 on chapter 16. This chapter we're looking at simple patterns of inheritance. So in part 1 we focused on Mendel's two laws of inheritance. We talked about the law of segregation and the law of independent assortment. In the second part of this chapter we looked at the chromosome theory of inheritance, so how all of this information is related to each other with the DNA, the mitosis, the meiosis, and these laws of inheritance and how they fit together. So in the last part of this chapter, we're going to look at a few extra things about inheritance. So we'll look at pedigree analysis very quickly. Pedigrees are pretty awesome. Unfortunately, we don't have enough time to spend on them. We'll look at X-linked inheritance, so looking at the sex chromosomes and how they behave a little. They have a little bit different things going on. A few really simple variations of inheritance, and then a little bit on the usefulness of genetics. So pedigree analysis, this is when you can look at an inherited trait and you can analyze it over the course of a few generations in one family. So this is really useful if you have a genetic disease that runs in the family, or if you're looking at the pedigrees of different animals and you want to see what's in their lineage or in their pedigree. So this is very useful in human genetics and also for agricultural purposes. We're going to be looking at humans. So cystic fibrosis is a disease where there's a lot of health problems. So these people, if you look on the little image here, they have a lot of sinus problems, trouble breathing, they have lung infections, enlarged hearts, a lot of trouble digesting food. So they have a lot of issues that they have to deal with if an individual has cystic fibrosis. This disease is a homozygous recessive disease. So individuals who are homozygous recessive are going to exhibit all of these symptoms of the disease. So what you can do is you can follow this disease through multiple generations. So this is a pedigree showing 3 generations. So females are represented as circles, males are represented as squares. Individuals that do not have the disease are white. Individuals that are affected, they do have the disease, are shaded in, so they're black. You can also determine if individuals are heterozygous, which means that they can potentially pass this trait on to their offspring. And those individuals are half white, half black, to show that they don't have the disease, but they do carry the gene for it. Okay, so here is a family where we see cystic fibrosis that runs in the family. So up at the top we have the grandparents. So we have a male and a female, and both of them are heterozygotes. And this is unfortunate for one of their children. So they have a female that does not have the disease, they produced a male that does have it, and then another female that is a heterozygote. So she can potentially pass it on to her offspring, which she did in the 3rd generation. So this is really useful because you can track the disease through lots of generations and kind of determine what's going on in these different lineages or in these different families. Other diseases, so cystic fibrosis is a recessive disease. Huntington disease is actually a dominant disease. So individuals that have this disease, they're probably going to pass it on to their offspring. This is set up the same way. So individuals that are shaded in have the disease. Males are squares, females are circles. So you can see that unfortunately this family has had a lot of Huntington disease within their family tree. And you can see that it started off with the grandpa who passed it on to his offspring, and then those parents passed it on to their children. So this is a really good tool to help families determine and to make family planning decisions. If you want to produce children, the chances of them having a certain disease that runs in the family, and kind of being ready to deal with that if that individual does end up having that disease. So the next, and again, like I mentioned, we didn't have a lot of time to spend on pedigrees. Hopefully, if you're really interested, you can kind of look at it on your own, because they are pretty neat to do and to understand. The next one is looking at X-linked inheritance. So sex determination in mammals, we use an XY system, but there's other types of sex determination in other types of animals. And we're probably the most familiar with the human one since we are humans, so we'll kind of stick with that, but just be aware that there are other types of mechanisms to help determine males and females from each other. In insects, the males have one X, females have two X's, so there is no Y in their genes. In birds, they use a Z and a W sex determination system. So males are actually ZZ, so they're homozygous, and the females are ZW, so they're heterozygous. So it's kind of the opposite of what we see in mammals, which is pretty neat. So it's kind of interesting to look and see how these different determinations for sex evolved over time. And then finally, in bees, males are haploid, so they have one copy of every chromosome, whereas females are diploid, so they have two copies of every chromosome. So that's pretty interesting as well. Another thing is environment. Environments, we see this a lot in reptiles and amphibians, so certain temperatures produce males, whereas a different temperature environment will produce females. And then hermaphrodites, these are when individuals have both male and female parts to them. But we'll focus on the male XY system, or the mammal XY system for our X-linked traits. So X-linked traits, in humans, the X chromosome is a lot larger and carries a lot more genes than the Y chromosome. Basically, the Y chromosome carries genes that help to code for male characteristics. The X chromosome, on the other hand, contains other genes that don't just have to do with gender or sex. So there's genes that are found on this X chromosome that are not on the Y chromosome. These are called X-linked genes because they're only found on the X chromosome. Because males are XY, they're said to be hemizygous for X genes. So whatever allele they get on that one X they have, that is the trait they're going to show. Because they're hemizygous, they only have one X. We're going to be looking at Hemophilia A as an example for our X-linked trait. So Hemophilia A is caused by a recessive X-linked gene, and hemophilia is a disease where your blood does not clot properly. So if you think about it, if you cut yourself or scrape your knee, you bleed and eventually it scabs over. Individuals that have hemophilia A, if they get a cut or they start bleeding, that bleeding doesn't stop, which you can kind of understand why that's bad. So if an individual gets into a really bad accident, a lot of times they will just bleed out because they're not able to control all of the bleeding. So if we take an individual, so if we take a male that is X with a big H on it, so they do not have the disease, their other chromosome is Y, and we cross it with a female that is X with a big H, and her other X has the gene for hemophilia A, so the little HA. So if we take those two individuals, we cross them together, we figure out their gametes, put it on a Punnett square, determine what types of offspring they can produce. So they can have a normal female, so XX with two big H's in the top left-hand corner. They can have a carrier female, so she has an X with the normal allele and an X with the hemophilia A allele. And she's said to be a carrier because she can potentially pass it to her offspring. And then if you look at the top right hand, we have a normal male, so he got the X with a big H, so he has the normal gene, and then a Y, so it's a male. And then the male in the bottom right-hand corner, this male is going to have hemophilia. And that's because he got the X that has the hemophilia A gene on it from his mother, and then his other chromosome is the Y, which makes him a male. So you can see that these two individuals, the chances of them producing an offspring that has hemophilia is 1 out of 4, or 25%. So you can kind of see that you can use this information. They can help them to make decisions if they want to have children. They have a 25% chance of having a child that has hemophilia. If they want to take that risk, are they ready to have that child and to deal with the consequences of a child with this disease? Hemophilia A is called the royal disease. If you have learned about the Russian Revolution and all in high school, you probably have heard of Anastasia. She had a brother who died of hemophilia A. And hemophilia A runs in some of the different royal families. And this is because a lot of times they do inbreeding, like they marry each other's cousins or other things like that. And this disease all came from Victoria, the Queen of England, up at the top. So here we have a pedigree showing hemophilia A in the different royal families. And I've labeled the English royal family on the left. There is Prince Charles, who we know a lot about, and then his sons are Harry and the one who just had the baby recently. Not too great with names, but you get the idea. So we have our English, or the British royal family on the left-hand side. We also have the German royal family, the Russian royal family. We see Anastasia. She was the youngest girl in that family. And then we also have the Spanish royal family as well. Individuals that have the red are either carrying the disease, so you see a bunch of females that are carriers, and then you see a lot of males that actually have the hemophilia A disease. So you can see that this disease spread through this royal family, just coming out from Victoria, the Queen of England, up at the top. Alright, so we looked a little bit at X-linked inheritance, and there's a problem in your packet that you can do for homework. We're going to quickly look at a few variations of inheritance, and then some usefulness, which I've already kind of talked about a little bit in the previous two parts of this section. So just to go over kind of what we've gone through already, so we've looked at something called Mendelian inheritance. So we looked at simple Mendelian inheritance in the first part of this chapter, where one trait is completely dominant over the other. So it's either like purple or white, or tall or dwarf, and one is dominant, one's recessive. We also talked a little bit about X-linked inheritance, where you have dominant recessive alleles, but they're only found in the X chromosome. So that is a little bit different, but you still follow Mendel's laws, except that you can determine if the offspring are male or female, and if they have the dominant or the recessive trait. So we're going to be looking at a few variations. So sometimes, instead of one trait being dominant over the other, neither allele is considered to be dominant, and this is called incomplete dominance. So when you get a heterozygote individual that has a copy of each allele, you're going to end up with some type of intermediate phenotype. It's kind of like a blending thing that occurs. So the classic example of this are pink 4-o'clock flowers. So how do we get, in this species of plant, you can have red, white, or pink flowers. So red flowers, you have 2 R's, so CR, CR. To get a white flower, you have CW, CW as your 2 alleles. In order to get a pink flower, you need a R and a W. So you can get this kind of blending between the pink and the red and the white. You end up with a pink flower. And this is because only 50% of the protein is being produced. So instead of getting 100% with red or 0%, you end up with white. 50% of that protein will give you a pink color. Another variation is multiple alleles. So each individual has 2 copies of every gene, or 2 alleles, but within a population you can actually have more than 2 alleles. So you can have 3 or more variants. So the phenotype depends on which 2 alleles you inherit. A good example of multiple alleles is the ABO blood type. So in humans, you don't have A blood or B blood. You can have A, you can have B, you can have O, you can have AB. So there's multiple alleles within the population that you can have. Another neat thing with blood type is that in order to get AB, this is called a codominance. So it's a little bit different than the incomplete dominance that we looked at, where we get kind of this blending or this intermediate phenotype. Codominance is when both of those alleles are expressed at the same time. So this is how you get AB blood. Another variation is sex-influenced inheritance. This is different than our X-linked traits. These traits are not on the sex chromosomes, but if you're male or female, depends, makes your allele dominant in one sex but recessive in the other. So a good example of this is male pattern baldness. So there's an allele for baldness. If this allele is in a man, it's dominant, so the man shows baldness. If the allele is found in a woman, it will act as a recessive allele. So only a woman that's homozygous for baldness would end up being bald, but in men, men just have to be heterozygous or homozygous dominant to become bald. And this is why more men go bald over women. So it depends on what gender you are and if this trait is dominant or recessive. So I think that's pretty neat to kind of understand why we see this certain characteristic show up in men more over women. And then a last variation is the role in the environment. So far we've been just talking about genotypes and how they relate to phenotypes, but the environment can also affect phenotypes. And this is called a norm of reaction. So the effects of the environmental variation on the phenotype. We see this a lot, and this is kind of that nature versus nurture debate. So how much of our traits and characteristics are based off of our nature, our genetics, versus our nurture or the environment in which we're raised. So here's an example of plants. So they took plants that were genetically identical to each other, so they controlled for the genes. They all have the same genes. And they just put these plants at different temperatures. And the plants grew at these different temperatures to different heights. So if you see on this graph, we have temperature on the bottom, which is our independent variable, the thing that we changed. So we planted plants at different temperatures. We then measured how tall these plants got, which is our dependent variable, which goes on the y-axis, up and down. So we find that at low temperatures, plants are shorter. We get to this optimum temperature where the plant grows the tallest, and then once you get too hot, the plants go back down in size. So this is showing that this environmental factor, the temperature, can affect how tall plants can grow. Another example, and this is in humans, this is a disease called PKU. This is a genetic disease, but it also depends on what's in your diet. And that will determine if you show the symptoms of the disease. So these 3 individuals, they're all siblings. They all have the PKU gene that will give them mental impairment and other disabilities. So the older girl in the wheelchair, she has it, the boy also has it, and then the little girl also has this gene. The only difference is that the oldest girl and the boy were fed just one type of diet, whereas the youngest girl was fed a special diet. And that diet gave her, or it limited certain things that would interact with the gene and cause the mental and physical disabilities. So this again is showing how diet, another environmental factor, can affect how a gene is expressed in different individuals. Alright, so we looked at our different variations of inheritance. The last thing I just want to mention is the usefulness of genetics, and I've kind of mentioned this a lot throughout this chapter, but just to give you it in words here. So genetics is used a lot in agronomy and also plants, and basically in any type of agriculture. So it's used a lot to protect offspring from crosses, to produce commercially important crops and livestock. So agriculture is all about increasing yield and figuring out how you can do that by crossing different types of plants or genetically engineering these plants. So this is one thing you can use genetics for. The other one is human genetics, which I've mentioned under the pedigrees, so following diseases in families, helping couples decide if they want to have children or if they don't want to have children based on different diseases in their families. So you can predict the probability of producing children with different inherited diseases if it runs in the family. So this is actually used a lot in family planning